should be fine.
Yes,
hello.
I can see you. Hello. So Dr. Francesco Biancardi did his master and then his executive MBA at the Politecnico di Milano. Then, driven by a strong passion for research and technological development, he started working in the field of light, electron, and X-ray microscopy, also interferometry, first as a specialist and then as a sales manager. Then, since 2017, his main goal is to support researcher and customer in implementing Zeiss setup microscopy. So the stage is yours, Francesco.
Thank you very much. You should see my screen now and the presentation. Please let me know if it's if you can see it correctly. So thank you for first of all for inviting us. As Anna introduced, I'm working in Zeiss with a special focus on, let's say, correlative microscopy, or what we like to say, how we like to say in recent years, beyond correlative microscopy. CLEM, as we saw just a few seconds ago, is a correlative light electron microscopy. It was born some decades ago to overcome the limitation of each microscopy technique, to see in more resolution with electron microscopy, and to check, for example, live imaging in light microscopy. One thing that is clear, and it's really clearer thinking about the talks that we heard before, is that the biological questions drive the technology. So we make technology, we make instruments to see basically what cannot be seen, and the biological question drive us to develop the right solution. And the second thing that is really interesting, especially thinking about the first speech, is that one size does not fit all, meaning that today's challenge of the research, so of your challenge, is by nature multi-scale, multimodal, multidimensional, multi-instrument, and multi-user. It's really difficult now to address one one scientific question that can be solved just with one source of information. And by source of information, I mean basically instrument. So what are we doing now is, as microscopy manufacturers, to integrate our wide portfolio, let's say, of instruments in one platform that integrates data. So each of these systems bring a different kind of data, and this must be put into context. The thing is, Zeiss by itself is a big company, is a multinational company with 34,000 employees. So we are quite big. We have a lot of products, but we don't cover all. We've just seen, for example, AFM that brings some information, and we do not make AFMs, but the data must be integrated into the context. So what we did, and here my colleague from Shanghai is showing, we can integrate the different scales in one single workspace, for example, and address localized information of different sources: wide field electron microscopy, fluorescence, confocal, 3D, and localize every information in a context, in a multi-scale context. So this is really important because when you think about different techniques, even pan-expansion microscopy, if you think, can bring some information that can be integrated in one interface. This said, everything must start with a very good image. And what makes an image great? Usually 3 points. Sorry, I'm going with the pointer, if you can see it. Sensitivity, to get the right information. Speed, especially if you need some dynamic information. And resolution, so the ability to distinguish the different two separate objects that are very close. The point is, all of these things apply to any technique, to every technique. So these things must be taken into consideration for every time you choose a microscopy technique. For example, here we have an example in Confocal. With modern confocals, you can reach 120 nanometer lateral resolution and 350 nanometer axial resolution with standard confocal imaging, which helps because you can use the standard preparation protocols. Preparation protocols are really important for every microscopy technique. This is especially true for super resolution. For super resolution, you can reach, you can have a wide range of resolution limits going beyond the diffraction limit, but you may be limited in preparation. So to get this result, you must be sure that you prepare the sample correctly, or you use the right resolution, super resolution technique. In this case, the lattice SEM, which is a Zeiss-specific technique for super resolution, basically lets you use any preparation protocol that is used for standard confocal imaging, but you have a better resolution and you have a wonderful live imaging. This said, not everything can be seen in fluorescence, and you may want to localize some, I don't know, bigger elements or, for example, organisms in a bigger scale. And here is where the X-ray microscopy, which is something new that I would like to introduce you, that maybe some of you have not heard so far, comes into play. What is X-ray microscopy? It's basically a computed tomography, so the CT that you find also in hospitals, but with the integration of an optical magnificator, which is basically a light microscope. So this is why X-ray microscopy. These are photons, but not in light, but in X-ray spectrum. The sample rotates to generate tomography, and the optical magnification allow you to reach resolutions that with a standard CT you cannot reach at all. Same is for the contrast. The interesting thing, you can see it with this tomography of a zebrafish. You can see the whole organism here in the full scale. The good point of the X-ray, of course, is that it's non-destructive. So the sample is completely intact after the analysis. What happens, you can get the tomography, the 3D tomogram of your sample, then decide, since it's a microscope, that you want to magnify a specific region, for example, this volume, and then you increase resolution and magnification with a higher numerical aperture objective. And here you have a tomogram at a higher resolution. For example, here you have 0.8 micron per voxel. If you think, you can apply this not only to zebrafish, but to any biological sample. The good point is that this system is so powerful, this technique is so powerful, is that you can actually see even single cells. Once you get the information on the single cells and you know where they are, then you can move to, and you select the event that you are interested in, then you move to the next technique that can be actually light microscopy. This is XRM. For example, here we want to see a neuron in a specific neuron that was stained in a specific way through different techniques. So light, X-ray, and finally ultrastructure, so electron microscopy information. Electron microscopy is still the technique of preference for to go to ultrastructural information because it's reliable. It has a standardized protocol that brings you the level of information down to the sub-nanometer scale. The important thing is that, as we heard before, technology is going on. So what was possible only with, for example, transmission electron microscope some time ago, today is possible with different techniques given by the so-called Gemini technology, that is the only one that allows to use a scanning electron microscope, for example, to do actually TEM-like imaging on ultrastructures. Other than other techniques, electron microscopy is really reliable for this kind of imaging. And The goal is to get, compared to TEM, for example, high contrast imaging like TEM, faster, with larger areas in an easier way, and not only to on thin slices like the TEM slices. What did we do? We are trying to get the same quality of image up to a certain point of resolution of TEM with the SEM. This is a challenge for you. Please write in the chat which is the TEM image here and which one is the SEM image. Just just one thing to share. One was acquired on a block, so a bulky sample on the top surface was acquired with the scanning electron microscope. Then the slice was cut and was put in the TEM, and was the same area was imaged with the TEM. One is the TEM, the other one is the SEM. Please answer which is which, and then we will collect. I can't see the chat, unfortunately. I don't know, Anna, if anybody is answering.
And not yet. I was trying to create a pool actually, but take a few minutes. I'm afraid that then I got lost.
Okay, I will. Let's take 30 seconds more. Take a look at this image again. One is TEM at 80 kilovolt, and the other one is the SEM at 1.5, if I don't remember, but kilovolt. So one is scanning and one is just take a guess or tell me what it is, because there is actually one thing for which you can actually say one is TEM and the other one is SEM. We can move forward because maybe no problem. But if anybody wants to tell, then we can go into it later. So no problem. The answer is on the left we have the TEM, and on the right is the SEM. So it was 1.9 kilovolts. This is another example. The idea is to get the same quality. You can see synaptic vesicles, neuron, mitochondria, and double membrane resolved even with scanning electron microscope. This allows, opens some interesting thing. You know that with TEM, to get volume, you need to combine several slices, which is quite complicated because the slice is very thin. It's 80 nanometer, more or less, usually, or 100 nanometers. If you want to have micron of volume, like in this case, where you reconstruct the neuron, you have to, it's impossible basically. So with this detector, with this technique, you can actually combine automated ultramicrotome, collect serial sections, and analyze them with this detector to combine the full volume. Or you can put the ultramicrotome inside the SEM chamber and have the reconstruction fully automated. You just align the blade and the beam and you reconstruct everything. Or finally, if you want the highest resolution, you don't go with a mechanical cut, but you go with an ion beam cut, so with ions, and then you reconstruct the same result. So 3 techniques to get the same, let's say, 3D correlative 3D volume approach. Why do we do this? For example, in array tomography, to reconstruct the big areas and reconstruct tomograms, so tomographies, at nanometer resolution with protocols that are recognized, of course, because you know that in this case it's a chemical preparation, chemical fixation, and substitution to get the ultrastructure, or with the integrated microtome. This is just an example. The ultramicrotome inside the chamber. How does it work? It works this way. You scan the block, you get your ultrastructure, then you cut the slice. This is a destructive technique, and this is why it could be very important to have in advance the X-ray microscopy technique, because you localize at a lower resolution where the event that you want to see, and then you go to the ultrastructure with this technique. And you have the reconstruction. Let's move forward. You know you are working with electrons. Electrons are basically a current, electric current. They are charged particles. The charged particles charge the sample. If you charge the sample, the beam that is getting to the surface is basically rejected, and you don't see anything. So you have to solve the so-called charging of the sample. How do we do that? There are two ways. One is you basically flood the chamber with positive ions. Positive ions take away the negative charge of the electron on the surface, but the problem is that you have ions and you have electrons running through air, because it's nitrogen ions basically. Electrons that run into air, they usually don't like it. They prefer to run in vacuum. So what was developed, and what actually you can see here, actually the difference between high vacuum imaging and so-called low vacuum, so nitrogen to compensate the charging imaging. You see the contrast and the resolution is much worse here. So what was developed and what proved to be really a game changer is a so-called focal charge compensation. Why focal charge compensation? Because you actually compensate the charging with ions flooded only on the area of interest. This way, you get this. You get focal charge compensation, even on samples that are really not conductive. Here is a rat spinal root image. You see the resolution, and you see on the right the 3D reconstruction of the axon, of the axons, very quickly. Hopefully, I hope you see the 3D movie. And here you then can play and segment, for example, the axons and reconstruct them, measure them, track them, and analyze them, whatever you want. The point is that you may want to get the image of the myelin, so the very high resolution. Let's move forward with some other examples. For example, mitochondria segmentation on 3D reconstructed, in this case, liver tissue. And how the final technique, the last technique that we can use for smaller volume but highest resolution is a focused ion beam. You basically cut a trench with the focused ion beam in red. It's ions that cut the sample, and you image with a cross beam, basically the cross electron beam that image the surface. You see here the reconstruction of the tomogram, and you get the same results. Some examples we saw before, for example, the presynaptic and postsynaptic analysis, let's say. Here with confocals, with standard confocals, you have poor resolution, and you can not distinguish if it's superimposed or encapsulated button, for example. With the resolution of the SEM, you can get it. So a work in this case made by EMBL some time ago basically mapped the opposed and not contacted, in this case, were synaptic buttons in a full volume. Just to show what the final result could be, this is the 3D reconstruction of the volume acquired with the FIB. So this is a tomography complete, and this is the segmented volume with neurons and dendrites and everything. In this case, the segmentation was made manually. You can imagine in this case the amount of work, because at the time there were not so many machine learning algorithms. Let's move forward. This is another example. You can see the volume is bigger. In this case, the full glia was reconstructed, and in this case, the acquisition was done not with the FIB SEM, so not with the ion beam, but with the serial block phase, so the ultramicrotome. Here it is. You can see the reconstruction of the glia and the neuron together. You can see the dendrite. The resolution is also here, the nanometer level. Finally, what if you want to image, get a full overview, for example, of a brain tissue? the goal is to reconstruct, to understand the mechanism that really drive for example, the memory of the brain, so reconstruct a bigger volume. For example, 1 cubic millimeter. If you want to reconstruct 1 cubic millimeter at the 4 nanometer resolution, it would take, only for the acquisition, 6 years. Then you have to segment the image. 6 years is basically 2 PhDs, may be a bit too long period. So what do? We need to use another technology. Again, the scientific question drives the technology. So we use multi-beam, multiple electron beams that scan simultaneously the sample and reconstruct in 1.3 seconds an image that would take hours with a standard electron microscope. Then you combine different images and you start to get terabytes of images. You need to then to think about how to handle this huge amount of data, to segment it, and to reconstruct at different scales the data that you want. This was done by Harvard recently and was acquired a 1.4 petabyte image, a full cubic millimeter brain tissue, reconstructed 130 million synapses. And you can actually browse it because it was made public. To handle this amount of data, there were basically a joint venture in some way between Harvard University, Professor Jeff Lichtman lab, Zeiss, and Google. In fact, you can find it in Google. Finally, and the last example that I want to show you about correlative workflows is the cryo workflow. Why cryo? The goal, again, the scientific question is we want to see our samples in the native state, because any other preparation protocol actually affects the sample. So we want to see the molecular interactions in the native state. How to do it? There is only one way. So vitrify, so freeze, but freeze in a certain way, the sample, and select the area of interest and go through multiple techniques. Which techniques? Cryo-Confocal. And then you need a confocal that is very gentle, not to melt your sample, because you must keep your sample at -200, more or less, -196. Then you go to cut a thin lamella, then to move to the TEM. So you locate the volume of interest. You cut the lamella, in this case, the cryo-fib. Here you see the lamella, very thin, and then you go to the TEM to locate the area volume, the region of interest, or the features that you want to see. Here you see cryo-Eriscan, so cryo-confocal, cryo-fib correlated in one workspace, you see, in the same space. And you have the resolution down to the nanometer level, typical SEM resolution. Then you go to the TEM. The TEM, where you want the TEM, you want the TEM when you want to go to the molecular resolution. Then you have no choice, basically, to see in the native state in a reliable way, of course. And this is the Cryo-Electron Tomography, where you see the different features, the superposition of the different data, ribosomes and even the proteins in the cells. And this is across all scales. So the idea is also to provide a way, a workflow and a method to connect the data. Let's say commercially, it's already there, and it's called Zen Connect. It's a connected workspace where you can put all the data. Here we saw only electron and light microscopy, but if you think you can integrate also AFM data, AFM provide data that are not reachable with other techniques. Think about the mechanical properties of a cell, for example. The good point, instead of working on different computers, different software, different data sets, and having difficulties in matching the information, here you bring the information into one single context and you navigate and basically analyze also with any segmentation tool, any machine learning tool, all these data. That's all from my side. I hope it was a little interesting, and I'm open for questions.